Hello friends, we are just waiting for the system to go live. Once I see myself on the YouTube, I'll start the discussion. There's maybe lag of few seconds between what I'm saying and what is getting transmitted to the YouTube. Just bear with me. As soon as the relay is ready on the YouTube, we will start the discussion, and I think it is live now. Great. So let us start our discussion for the biochemistry. Now you know biochemistry has become very important. You're getting lots and lots of questions. Some applied, some concept based, and overall focus is on mostly the basics. So if your basics are clear, then you should be able to come out with flying colors. Now biochemistry is one subject where you can score very high, because this is something where you know or you don't know. There is no middle ground, unlike the clinical subjects where you have to apply a lot of things and then you have to reach the conclusion. Biochemistry is something where you can definitely be sure about your answer or you don't know the answer. There is no middle ground or confusion sort of things are not there. Okay, so good evening everyone, and we'll be looking at some very important concepts. I'll be using a few questions also to give you the concepts. For the molecular biology aspect, I'll rather want to highlight the points which you should be knowing rather than individually talking about the questions, because that is something where every time we get a different sort of question, sometimes very concept based, sometimes very technique based. So there, rather than talking about questions, I'll take a few minutes and quickly highlight the important specific points which are likely to land up in the exam. So for our discussion, I will start from the very important, very commonly repeated, and the very basic point which all of you must know. Very easy to crack the question, provided you pay attention to the very simple things. Now if you look at this question, this is very troubling because it is a big question. The options are also very large, and it seems it is going to take a lot of time. It is a tricky question, but there is a very simple trick to solve this question which we'll try to follow. Okay, just for the record, I'll read out the question. Following is a result of ABG analysis. So first point which you have to know here is, what do you mean when we are saying ABG analysis? I'm sure all of you know ABG analysis means we are talking about the arterial blood gas, right? We are talking about the arterial blood gas analysis, and whenever we say ABG, immediately your mind should go to the acid-base imbalance. ABG analysis, we are looking for the acid-base imbalance. Now, when we are talking about the acid-base homeostasis, there the three important points which you have to know. What is the value of pH? What is the value of PCO2? And what is the bicarbonate concentration? So typically in the question you'll see the value of pH is given, the value of PCO2 is given, the value of bicarbonate is given. Based on these three, you have to make a diagnosis. Now when you're making the diagnosis, there are two parts in that diagnosis. First, the primary cause, and second, whether the compensation is there or not. If it is there, what is the type of compensation? So this is something which we have to answer. Almost always we get a question from this particular topic. So very quickly I'll tell you how to attempt this question and what will be the answer to this question. So when we're talking about acid-base imbalance, what you have to know, the normal pH is 7.4, the range being 7.35 to 7.45. I'm sure all of you know this. This is mostly a recap that we are doing. Second, when we talk about, I'm so sorry. Second, when we talk about, okay, we are talking about the pH, and second, we talk about the PCO2. When you talk about the PCO2, in case of PCO2, again, you know, the value should be 40 millimeter of Hg, 40 millimeter of Hg, and again, the range can be somewhere between 35 to 45, right? Lastly, we come to the bicarbonate concentration. In this case, typically the range should be 24 milli equivalent per liter. Okay, and the range can be anywhere between 22 to 26 milliequivalent per liter. These are normal values which we are expected to know. Almost always they will not give these values in the question, so you're expected to know that these are normal values when we are talking about acid-base discussions, and these values relate to the blood. We are talking about the blood here. Now there are two possibilities. Either you can have acidosis, right, or you can have alkalosis. These are the two possibilities that we will come across, either acidosis or alkalosis. When we say acidosis, what does it mean? The value will be less than 7.4. When we say alkalosis, the value will be more than 7.4. So based on value, immediately we can determine what we are talking about, whether it is acidosis or alkalosis. So one point of the diagnosis is clear. Look at the pH value, we know whether it is acidosis or alkalosis. Once you determine whether it is acidosis or alkalosis, the second part that you have to determine is what is the cause. So if we talk about the PCO2, the PCO2 is going to contribute the H plus ions. So when we talk about acidosis, more PCO2, more H plus will be there. So if PCO2 is the cause, then PCO2 should be increased. Only when it is increased, more H plus will be there and acidosis will be there. If we talk about the bicarbonate concentration, if bicarbonate concentration is affecting the acidosis, affecting the pH, in that case the bicarbonate value should fall. If it is less, again it will cause acidosis. So PCO2 changes are respiratory in nature. Bicarbonate changes are metabolic in nature. Sometimes they can also be called renal in nature. So in acidosis, either PCO2 is increased, in which case it will be respiratory, or bicarbonate is decreased, in which case it will be metabolic in nature. Similarly, for alkalosis, what will happen to PCO2? PCO2 should be decreased so that we have less of H plus and alkalosis will be there, or the bicarbonate concentration should be increased. So number one, look at the pH. So we know whether it is acidosis or alkalosis. Based on the result of pH, look at the values of PCO2 and bicarbonate. So if you're talking about acidosis, if PCO2 is increased, then it is respiratory acidosis. If bicarbonate is decreased, then it is metabolic acidosis. So this is part 2 of the diagnosis. So respiratory or metabolic, acidosis or alkalosis. The last part I told you is whether the compensation is there or not. Now what about compensation? So if we are talking about acidosis and PCO2 value should be increased, in that case there are three possibilities when you look at the values. It is increased, it is normal, or it is decreased. If it is increased, then it is primary cause. If it is normal, then no comments. And if it is decreased, in that case this is the compensation. So in acidosis, PCO2 should be increased, but if it is decreased, then it is compensation. Similarly, in alkalosis, PCO2 should be decreased, but if it is increased, then it is the compensation part. So this is what you have to keep in mind. You look at the result, you make some predictions, you check the values. The value which matches the prediction is the primary cause. The value which goes against your prediction is the compensation. So based on that, you can quickly make the diagnosis. I can see a lot of you have sent in your answers. If we go back to the question, so here what we can see, acidosis is there. You can see pH is less than 7.4. Acidosis is there. In acidosis, PCO2 should be elevated. We can see PCO2 is elevated. Bicarbonate should be decreased, but here what you find, the bicarbonate is actually increased, meaning this is the primary cause, this is the compensation. PCO2 is relating to the respiratory system, So respiratory acidosis. So this is out, this is out. These are the two options. And second, the bicarbonate will relate to metabolic. So metabolic compensation. Respiratory acidosis with metabolic or renal compensation. So this will be our answer. Respiratory acidosis with the renal compensation. I hope this is clear to everyone. How to attempt this question? This question will land up again and again and again. I'll show you some more questions which have come from this topic. Every time they want to know whether you know the acid-base homeostasis or not. Why? Because when you're looking at the patient in the ward, invariably any long-term patient, post-surgical patient is going to have some acid-base disorder. Imbalance will be there, so you should be able to identify, and subsequently only you will treat. If you are not able to identify the acid-base disorder, how are you going to treat? So that is why always we get this question. So I'll quickly summarize. See here, we get the arterial blood sample, we look at the pH value. If it is less than 7.4, then it is acidosis. In acidosis, the bicarbonate should be decreased. If it is decreased, then it is metabolic in nature. And if PCO2 is going to lesser value, it should be increased in acidosis. In that case, it is compensation. On the other hand, if respiratory is the primary cause, PCO2 value should be more than 40, more than normal. And in case of compensation, the bicarbonate rather than decreasing should increase. So you can see here, if primary cause, then decreased. If compensation, then increased. Similarly for PCO2, primary cause increased. In compensation, it will decrease. Going to the alkalosis part, pH value more than 7.4, alkalosis, bicarbonate value should be higher in metabolic alkalosis, and the PCO2 value should also be higher in compensation. The PCO2 value should be less in respiratory alkalosis, and renal compensation, if it is there, the bicarbonate will also be less. So this is a very simple flow chart which can help you in identifying what is the primary disorder and whether the compensation is there or not, whether the compensation is there or not. Okay, so you make a prediction for the PCO2 and the bicarbonate based on whether it is acidosis or alkalosis. If your prediction is wrong, then compensation is there. Okay, prediction is wrong for PCO2, then the respiratory compensation. If prediction is wrong for bicarbonate, then renal or the metabolic compensation is there. Okay, so your prediction will help you identify the primary cause as well as the compensation. So, okay, let's see, we have some. Okay, this is Davenport diagram. Sometimes they can give you this and they can ask you what is this. So what is the utility of Davenport diagram? It can be used in the emergency rooms to quickly identify the type of acid-base disorder. If you're not very sure about your interpretation of acid-base disorders, this can be a handy help or guide. Most of the interns would want to have this when they are placed in the emergency, because you just put in the values, bicarbonate value, pH value, the PCO2 value, and immediately wherever the dot falls, based on that you can identify the type of acid-base disorder you are looking at. This is known as the Davenport diagram. So this is another likely question that might land up in the exam. Okay. Okay, so I'll show you some more questions which have come in the exam. See here, the lab report of a patient gives pH value 7.2, acidosis. Bicarbonate is decreased, which should be there in acidosis. So this is probably metabolic. PCO2, if it is acidosis, then it would have increased. But here it is going below. So it would be compensation. However, in the question, compensation has not been asked. So we are looking at metabolic acidosis. Your answer in this case should be metabolic acidosis. Okay. Lastly, sometimes they can expect you to know some of the common causes for the metabolic acidosis, because this is clinically very important. Very frequently in the clinical setup we are going to find the metabolic acidosis only. So another question, take a look. 55-year-old male presents with tachypnea and mental confusion. Blood glucose level is 350 milligram per deciliter. pH is 7. pH is 7 means we are dealing with the acidosis. Urine shows presence of ketone bodies. What is the most likely acid-base disorder? So here the expectation of the examiner is you should be able to tell us that diabetic ketoacidosis is responsible for the metabolic acidosis. We can quickly know that this is a case of decay, right? Blood sugar is elevated, ketone bodies in the urine, pH is decreased. So this is a case of diabetic ketoacidosis, which is a type of metabolic acidosis. So this is how the acid base is landing up again and again in an exam. Just by knowing the basic concept, we should be able to sail through this question. Easy, 4 marks for you. Very good, very good. So let us quickly move on to another very important concept which you should know about, and that is the enzyme inhibition. Every time they're talking about enzyme inhibition. Why they're asking about enzyme inhibition? Because this relates to the drugs that you're going to use in pharmacology. So if you don't know the enzyme inhibition, you might not be able to interpret the action of the drugs or how they are causing the interaction in the body, whether an intermediate is there or not, if there is poisoning, if there is overdose, how to overcome that overdose. All that is based on the mechanism of inhibition, where the most common, most important type of inhibition will be the competitive inhibition. So sometimes they'll give you the diagram, they'll ask you what is the type of inhibition. Sometimes they'll give you the inhibition, they'll ask you what is the change in the Km and Vmax. These are the two parameters which are used to identify the type of inhibition. Sometimes they'll give you a graph and they will give you the drug. They'll ask you which drug among the given options follows the inhibition shown in the graph. So that is a two-part question. First you have to identify the type of inhibition, then you have to know which of the drugs is following that type of inhibition. So let me quickly tell you the type of reversible inhibitions. We'll also take a look at the changes in the graph in the Lineweaver work plot, and very quickly we'll come back to the question and give the correct answer. Alright, just a second. Okay, I can see most of you have tried to give the answer, and a large number of the answers are even correct. Okay, let us see what happens. So when we are talking about the enzyme inhibition, first thing that you have to remember that enzyme inhibition can either be reversible, which is clinically very important, or it can be irreversible. In case of irreversible, the subtypes are not important because management for all irreversible subtypes is still the same. So when you talk about the reversible enzyme inhibition, the three subtypes are competitive, the non-competitive, and the uncompetitive. So here there are three major differences when you compare the different types of reversible inhibition. The differences are in Km, the Vmax, and where the inhibitor attaches to the enzyme or the enzyme substrate complex. These are the 3 points of differences that we see when we talk about the different types of the enzyme inhibition. So if you look at the Km and Vmax for the different types of inhibition, what you will find that Km is increased for competitive inhibition, Km is normal for non-competitive inhibition, and Km is decreased for uncompetitive inhibition. Okay. Whereas when we talk about Vmax, Vmax is simpler to remember. It doesn't change in competitive, but it is decreased in both non-competitive and uncompetitive. The last point that I told you about point of attachment, what you have to remember, in case of competitive, the point of attachment is the active site. The point of attachment is the active site. In non-competitive, the point of attachment is, the point of attachment is active site. In case of non-competitive, the point of attachment is the allosteric site. In case of uncompetitive inhibition, the point of attachment is enzyme substrate complex. right? So until unless the enzyme substrate complex is formed, the inhibitor will not be able to act. The second part of this enzyme substrate inhibition, that you have to know is the changes in Line-Weaver-Burke plot. Line-Weaver-Burke plot is used to study the inhibitors and compare with the non-inhibited reaction, because in Line-Weaver-Burke plot we get the straight line. In Michaelis-Menten we get the curved line, so their comparison is difficult. Here comparison is much simpler because we are getting the straight line graph. So when you look at the blue lines are the uninhibited graph. So it will be same in all the three graphs. The red line is the inhibited graph. So you can see the different types of graph are there. About the Line-Weaver-Burke plot, what we have to know, that where we are crossing the y-axis, that point is 1 upon Vmax. Where we are touching the x-axis, that point is minus 1 upon Km. Okay, and now I'll give you the trick to identify the type of inhibition in the graph. How we are able to identify which type of inhibition are we seeing in the graph, because sometimes when we are in the rush, when we are in the pressure, it can become confusing. We might not be able to quickly apply our mind. So I'll give you the trick. The trick is in this table which is there at the bottom. What you have to remember is what doesn't change. What doesn't change will help you to identify the type of inhibition. So in competitive inhibition, the Vmax doesn't change. In non-competitive inhibition, the Km doesn't change. Whereas in uncompetitive, there is no unit which is unchanged. Both Km and Vmax will change. Once you know this, and you know a little bit about Line-Weaver-Burke plot, and now you see the graph, then it is very simple. In graph 1, you see both the graphs, both the lines are passing through the same point on the y-axis. On the y-axis, what did we say, what is there? The Vmax is there. Meaning in the uninhibited reaction and the inhibited reaction, Vmax is unchanged. Unchanged Vmax where? Unchanged Vmax where? Competitive inhibition. In the last graph, if you see, the point on x-axis is the same. What is on x-axis? Km. So Km is unchanged for the non-competitive inhibition. So when you see the same point on the x-axis, it is non-competitive. And lastly here, no common points. No common points means we are dealing with the uncompetitive inhibition. So from graph now, even if you don't know what is increasing, what is decreasing, just remember what is not changed, and quickly you can identify the type of inhibition being shown in the graph. I hope this point is now clear, how to identify the type of inhibition from the graph. Now I'll repeat, the competitive inhibition is the most frequently asked question in the exam, because a large number of drugs that we're using clinically will interact with the enzymes via the competitive inhibition. Okay, so you must know what is the appearance of competitive inhibition on the Line-Weaver-Burke plot, what are the changes in Km and Vmax in case of a competitive inhibition. Okay, so these are some of the points which you have to keep in mind when we are talking about the reversible type of enzyme inhibition. Please note, all these three are a reversible type of the enzyme inhibition. Okay, so if we go back to the question that we had, okay, true about competitive inhibition. The first point we remember is Vmax remains the same. Luckily for us, in only one of the options the Vmax remains the same, that is option B. You don't even need to comment on the Km. And now let's look at the Km. Km increases, very true, for the competitive inhibition. So this is how you can quickly answer the question. Okay, Km remains same, Vmax decreases, rightly said. This is the non-competitive inhibition, whereas Km decreases, Vmax increases. We don't see this Vmax increase, we don't see with inhibitors. Obviously, we don't see the increase in Vmax with inhibitors, they will rather be activators. Okay, so that was about the enzyme inhibition. Second very important question which is coming nowadays from the enzyme inhibition is about the catalytic efficiency. Actually, the Km and Vmax are used to monitor the catalytic efficiency only, but there is also a formula to calculate the catalytic efficiency, and now the examiners want you to know the formula for catalytic efficiency, which will be the Kcat divided by Km. Both are constants, so you need not know go into the details of what exactly are Kcat and Km. They are constants which are described for the given reaction. So based on the value, sometimes you can be asked to do the calculation, or in one of the recent exams they asked which among the following is actually the catalytic efficiency parameter. So you should know the formula. Catalytic efficiency is the ratio between Kcat and Km, and then you should be able to do the calculation. Now you'll say, oh, what is there in the calculation? We can quickly calculate and compare the four values. So the problem in doing the calculation is now we have forgotten all the physics that we used to do in 11th and 12th when you're preparing for the entrance exam for the UG entrance, right? The very important thing to keep in mind when you are doing any calculation based MCQ, what is that important consideration? That important consideration is units. You have to keep in mind that units for all the four options are similar before you do the comparison. So that is where the examiner will catch you, and that is what had happened in this question. If you can see, micromole, 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 but here it is nanomole, right? So when you do the calculation and you don't notice the nanomole and you use this value of 200 for doing the calculation, your answer is wrong. Alright, so please note, first point, we have to make sure that the units in all the option is the same before you do the comparison. This means 200 nanomole is actually 0.2 micromole. 200 nanomole is actually 0.2 micromole. And now when you do the calculations, what will happen? This value will become 50 divided by 0.2, which is 250, and this will come out to be the highest value. This was the trap that the examiners had put for the students and us, and a large number of students definitely made this mistake of ignoring the units. I'll repeat, not only for this question, for any other question where calculation is involved, you have to make sure that the units are same. If they're not same, do the conversion to make the units same. Only then you can do the comparison. Before that you cannot do the comparison. Okay, before that you cannot do the comparison. Second aspect, make sure you write the formula correctly. If you write it upside down, then your answer will obviously be wrong. So you have to make sure that you're writing the formula correctly before you do the calculation. See, in the question, the second part is they've given the value of Km first and the value of Kcat later, whereas Kcat is in the numerator and the Km is there in the denominator. Alright, so please note, the units have to be same before you can do the comparison. Let's move on. Now another very simple, very basic question which you have known from your school days also, and that is RQ. So a person is of 50 kg weight, they've given weight. His CO2 and O2 levels were measured from spirometry. Dr. Purval sir would have told you, or if you see his videos, there he will talk about spirometry. So CO2 value and the O2 value is provided. What is the RQ in this case? Okay. So very simple. What you have to know here, the value of RQ is the amount of CO produced divided by the amount of O2 which is consumed. Okay, the amount of O2 consumed should be in the denominator. The CO2 which has been produced should be in the numerator. Whatever value you get is the RQ or the respiratory quotient. Okay, so you can see here the value of weight is just to confuse you, so that you make some erroneous calculations and reach to the other options. So if you put in the values correctly, it will be 200 divided by 250 to give you a value of 0.8, which is option C. Very good. Second part, sometimes they can directly ask you on the RQ for the different macromolecules. So you should also know the RQ values for the different macromolecules, which I'll highlight here. For the carbohydrates, the RQ is 1. For the proteins, the RQ is 0.8 to 0.9. For the fats, the RQ is 0.7. Two additional RQ which you should know. Alcohol has a very high respiratory quotient. Alcohol has RQ of 0.66. Lastly, if absolutely anaerobic respiration is going on, in that case the value of RQ will be infinity. But if a small contribution is coming from the anaerobic respiration, in that case the value of RQ should be greater than 1. So if you get the value of RQ greater than 1, what this means, there is at least some amount of anaerobic respiration. At least some amount of anaerobic respiration is ongoing if the value of RQ is more than 1. You can see for most of the dietary stuff, carbohydrate, protein, the fat, the alcohol, the value is less than 1. It will go to more than 1 only when some anaerobic respiration is going on. So what will you have to remember? First, what is the formula? How to do the calculation for RQ. Second, what is the RQ for the important food stuff? Third, what happens in anaerobic respiration, and what is the result on total RQ if some anaerobic respiration is ongoing inside our body. Okay, so very simple, very important, very basic points which you have to keep in mind when you get the question on RQ. Okay, so like most of you said correctly, the RQ in this case would be 0.8. Okay, now this is a very important question, and very concept based question, in the sense that if you don't know the fundamentals, you will not be able to answer this question. I'll read the question with you. A young man met with a motorbike accident and had injury to ileum and jejunum. You can see it is one of the recent questions. Therefore, the entire ileum and partial jejunum was resected. Please note, the ileum and the jejunum was resected. Okay. Okay. So what is happening here? Which of the following is the patient most likely to suffer from? So I can see a lot of answers coming in. Before we go to the answer, I just like to mention a few things about this question. I'm so happy to see that most of you are able to identify the problem here. So the very important concept about vitamin B12 is, unlike other vitamins, different zones of the digestive system are involved in the absorption, whereas for the other vitamins, one zone would be responsible for the absorption, but here multiple zones are responsible for the absorption. So how come? The oral mucosa will give us the copalophyllum. Okay, so one output from the oral mucosa, if there is decreased secretion from the oral mucosa, if there is keratoconjunctivitis sicca where secretion is not coming from the mouth, when there is dry mouth, the factor will not be there. Then from gastric mucosa, you get the intrinsic factor. If gastric mucosa is not doing the secretion, then intrinsic factor will not be there. Atrophic gastritis is there. If the gastric resection is there because of any cancer or because of any reason the stomach has been removed, in that case intrinsic factor will not be there. Lastly, absorption occurs in the ileum. So because of any reason, if you remove the ileum, the absorption will not occur. So please note, all three should be intact. They should be functionally active. So they should be intact, they should be functionally active. If any of the three components is not working, if it is not available, the B12 absorption will not take place. This is a very, very important concept. In one of the exams, we had two questions talking about absorption, involving the two different aspects of the B12 absorption. So B12 absorption is unique among all the vitamins, and therefore it is almost always asked every alternate year. You'll get the question related to the B12 absorption. And second vitamin where we commonly get the question is vitamin D. This is one of the two vitamins which can be synthesized by the enzymes in the body. The second one being the vitamin B3. Okay, so B3 and vitamin D can be synthesized by the enzymes within our own body. Some additional vitamins can be synthesized by the gut bacteria. Okay, so you should know how the vitamin D synthesis is taking place in our body, and you should also know the sequence of events in the vitamin D synthesis. So very quickly we'll do that sequence of events and then come back to this question. Oh, for the previous question, I think I didn't give the answer. The answer was the vitamin B12 deficiency, as we discussed, because the ileum has been resected. You can see entire ileum has been resected, so the absorption will not take place. Okay, so here you have to identify what is the correct order of organs which are involved in vitamin D metabolism. So first and foremost, the synthesis of the vitamin D occurs in the skin. The cholesterol molecule, under the influence of the UV part of the light, will get converted into the pre-vitamin D, right? This pre-vitamin D will quickly isomerize to give us the vitamin D3. This is the precursor form of the active vitamin D. The D3 is coming from cholesterol. Now, sometimes you can get the vitamin D from the diet. If the source of the vitamin D in the diet is animal, in that case you get the D3. Whereas if the source of vitamin D is plants, in that case you get vitamin D2. Ultimately they will give rise to the similar functional end product, but some difference in structure is there, based on which we have the terms the D2 and D3 for the vitamin D coming from the plant and the animal source. So in our body, the vitamin D which is synthesized is vitamin D3. In our food stuff, if the source of vitamin D3 is the animal food, animal product, then it will be the D3. If the source of vitamin D is the plant product, in that case it will be the vitamin D2. Whether it is synthesized in the body or it is coming from the diet, whether it is from plants or animals, subsequently the same set of reactions will take place, which are: first, the vitamin D3 in the liver will be acted upon by the 25 hydroxylase. So what you get? 25 hydroxycholecalciferol. What you get is 25 hydroxycholecalciferol. You can write it as 25 hydroxy D3, or if it is D2 you can write it as D2. 25 hydroxy D3 or D2 will be the first product. As you can see, this conversion is occurring in the liver. So first was skin. After skin we go to the liver. Subsequently the 25 hydroxycholecalciferol is acted upon by the 1-hydroxylase. Okay, and the 1-hydroxylase will now convert it to 125 dihydroxycholecalciferol. So you can write it as 125 dihydroxycholecalciferol, whether it is D3 or D2. Accordingly you can write the suffix that D3 or D2 is there. So to summarize, skin, cholesterol conversion to D3, liver, conversion to the 25 hydroxy form, and finally in the kidney where it is the 125 diadoxyform. The 125 diadoxyform is the functional, or you can say metabolically active vitamin D3. Please note that 125 dihydroxy is quickly inactivated by the 24 hydroxylase. It has very short half-life, so normally in the blood we don't measure the 125 dihydroxycholecalciferol. What we measure is that 25 hydroxycholecalciferol, which doesn't readily undergo conversion. Why? Because this hydroxylase is there in the kidney. Again, the 24 hydroxylase is also there in the kidney. So this product from the liver can stay in the circulation for longer time. Only when it reaches the kidney it will get inactivated. So to measure the vitamin D level, which form of vitamin D is estimated? Is it the D3 form? No. Is it the 125 form? No. 25 hydroxy form is measured to estimate the level of vitamin D in the body. Okay. Only when we find adequate vitamin 25 hydroxy D3 and still we find signs and symptoms of the vitamin D deficiency, we would like to measure the 125 dihydroxy form to make a diagnosis which is typically genetic in nature, whereby the conversion to hydroxylase is either not occurring or there is resistance in the receptors. To make that distinction, we have to measure the 125 hydroxy form. If the 125 dihydroxy form is decreased, then the problem is in the 1-hydroxylase. But if the 125 dihydroxy form is adequate, then the problem is in the receptor, in which case we call it the vitamin D resistant rickets. Okay, so the treatment for that will be different. It is mostly symptomatic, we can't do much. But if the 125 dihydroxy form is decreased, there you can give the supplement of the active form vitamin D, where the response will be seen. Okay, so these are the points in vitamin D, the different aspects which will be asked in the exam, about which you should know the details. Now another very simple, very straightforward question and very frequently asked question in the exam. Okay, so again I'm so sorry I didn't give the answer for the question. So skin, liver, kidney, this is the sequence that we are going to use when we are synthesizing vitamin D in our body. Vitamin D metabolism involves the skin, liver and kidney in that sequence. Okay. Question number not important. So we are talking about the glucose transport. Insulin mediated glucose uptake in myocytes is affected by, and I can see lots and lots of answers coming in very quickly. Students are sending in the answers. Okay, so rightly said, most of you have given the correct answer. The GLUT4 is present in myocytes and adipocytes, where the absorption is insulin mediated. So let us quickly take a look at the different insulin transporters and where they are located, what is their important role, and what are the points you should know about them. Okay, let's see. So when we talk about the insulin transporters, what we have to note, number one, we have two types, the GLUTs and the SGLT, right? The GLUT and the SGLT. So GLUTs are facilitative and bidirectional, whereas SGLT is sodium dependent and unidirectional. Okay, these are the points which you have to keep in mind. When we talk about the GLUT series, we have till now 14 different forms have been identified. We don't need to know the details about all of them. If you know about the first 5, that is more than enough. Similarly, for the SGLT, we have type 1 and type 2. Details of 1, if you know, is enough. Please note, for the SGLT2, now we have the inhibitors which are available, which are used in the treatment of the diabetes mellitus. Inhibitor of SGLT2 used in treatment of diabetes mellitus. SGLT2 is present in the renal cells. Okay, in the tubular cells, in the renal epithelium, these SGLT2 are there. So if you block them, the reabsorption through the SGLT2 will be inhibited. Okay, that is additional point. So about the GLUT1, most important thing, it is present everywhere. You can see it is present everywhere. most of the tissues will have another transporter in addition to GLUT1, except for the RBC. So in RBC, the only type of transporter which is available is the GLUT1. As you can see, it is responsible for the basal transport of the glucose. The GLUT2 is present at very specific places. It is present in the liver, the beta cell of pancreas, the small intestine, and in the kidney. It is responsible for rapid uptake or release of the glucose. GLUT3 is present in the brain, kidney, placenta. It has lowest K, means it has highest affinity. It is able to extract the glucose very quickly from the low concentrations. And the GLUT4 is an insulin stimulated glucose uptake present in the muscles and the adipocyte, GLUT5 present in the small intestine. It is responsible for absorption of fructose from the luminal side. The GLUT5 is not present in the renal tubular cells, so the kidney cannot reabsorb fructose. The intestine can absorb fructose, but once fructose is filtered in the filtrate at the level of kidney, it cannot be reabsorbed. Why? Because the main transporter there on the tubular side is SGLT1 in the small intestine and kidney, which does not take up fructose. It only takes up the glucose and the galactose. It will take up only the glucose and the galactose. And the question which comes is about the transporters in the intestine. Which transporter is there on which surface? So you can see here SGLT1 and GLUT5 are there on the luminal surface, whereas the GLUT2 is present on the basolateral surface. On the basolateral surface, we have the GLUT2. Okay, similar arrangement is there in the kidney. So only thing you have to remember that in kidney we don't have the GLUT5. GLUT5 is not there in the kidney, so the absorption of fructose from the luminal side will not occur. Okay, another very important question, very frequently asked in your exam, will be the key enzyme of the gluconeogenesis. Gluconeogenesis means new glucose synthesis. So if we look at the process of gluconeogenesis, and it shares several enzymes with the process of glycolysis, but some enzymes are unique to each of them. So in this case, they are asking about key enzyme of gluconeogenesis. Let us first enlist the enzymes which are specific for the gluconeogenesis, and then we'll identify the key enzyme. Okay. So let us quickly take a look at the enzymes. For glycolysis, there are three enzymes which catalyze the irreversible reaction. We know that the three enzymes which catalyze the irreversible reaction, the hexokinase or glucokinase, the PFK1 and the pyruvate kinase, wherein the PFK1 is the most important rate limiting enzyme, right? It is the most important rate limiting enzyme. On the other hand, if we talk about gluconeogenesis, the action of pyruvate kinase is bypassed using the pyruvate carboxylase and the PepCK, phosphoryl carboxykinase. The PFK1 action is bypassed by fructose 1,6-bisphosphatase, and the action of hexokinase is bypassed by the glucose 6-phosphatase. Please note the rate limiting enzyme is fructose 1,6-bisphosphatase, meaning this will be the most important enzyme or the key enzyme of the gluconeogenesis. Most important enzyme of the gluconeogenesis will be fructose 1,6-bisphosphatase. This is identified as the rate limiting enzyme of the gluconeogenesis. Okay. Please note this comparison is asked again and again and again. You should be able to identify the enzymes which catalyze the irreversible reaction in glycolysis and the enzymes which catalyze the irreversible reactions in the gluconeogenesis. The remaining enzymes for the two pathways are common. They are same because they are catalyzing the reversible reaction. Now, second follow-up question from here is very important about the insulin glucagon ratio. What is happening in case of insulin glucagon ratio? Now again, this is a part of the series of questions which you should know what is happening in case of the fed state and the fasting state. What is happening when there is increased insulin? What is happening when there are changes in the insulin glucagon ratio, right? And lastly, what happens, which enzymes are active in the phosphorylated state and which enzymes are active in the dephosphorylated state. These are four different scenarios that you will commonly get when we are talking about the enzyme action in terms of the fed state, fasting state, insulin glucagon ratio, and phosphorylation, dephosphorylation. So what you have to remember, very simple. When glucose level increases, in that case the insulin will increase. When glucose increases, the insulin increases. Purpose, it will try to decrease the level of glucose, right? So in increased insulin means we are talking about the fed state. They are one and the same thing. Okay, talking about the insulin glucagon ratio. Now you know that insulin and glucagon have a reciprocal relationship, right? If insulin level will rise, the glucagon ratio will fall. If the insulin level will fall, the glucagon amount will rise. So let's see, if there is increase in insulin, the glucagon will decrease, right? If there is an increase in insulin, the glucagon will decrease, meaning overall the ratio will increase. The numerator has expanded, denominator has decreased, so the value of the ratio will increase. On the other hand, if the insulin decreases, the glucagon will increase. The numerator has decreased, denominator has expanded, so what will happen? The ratio will decrease. So what you are seeing here, if insulin increases, the ratio increases. If insulin decreases, the ratio decreases. So whenever you get the question on the insulin glucagon ratio, you can very quickly ignore the part where it mentions the glucagon ratio. Change in insulin will increase the ratio. So if it says the ratio is increased, means insulin is increased. If it says the ratio is decreased, it means the insulin is the component which is decreased, and we interpret accordingly. Last part which you have to remember is insulin will mainly cause dephosphorylation. There can be certain exceptions, but as a general rule, what you have to remember, insulin will cause the dephosphorylation, right? So what we have to know now, very simple, what is the action of insulin? Because all the four questions are finally depending on insulin only. So which pathways are activated by insulin, which pathways are down-regulated by insulin? There only we'll get the answer that which enzymes are getting activated, which enzymes are getting inactivated, which enzymes are active in the phosphorylated form, which enzymes are activated in the dephosphorylated form. So let's see. Action of insulin. Before going there, action of insulin is again very simple. It will decrease glucose, right? We already said that it is going to decrease glucose. So any pathway which is consuming glucose will be upregulated, right? Second, insulin rises when glucose is abandoned. Glucose is the preferred fuel. So second part is it will decrease production of fuel. So if there is any pathway which is generating the fuel, whether it is glucose, whether it is fatty acid, whether it is ketone body, they will be decreased. So if you remember this, then the picture becomes very clear. Glycolysis uses glucose, glycogenesis uses glucose, lipogenesis uses the energy from glucose, HMP shunt uses glucose, amino acid synthesis uses glucose, cholesterol synthesis will use acetyl coenzyme coming from the glucose. So all of this will decrease the amount of glucose. On the other hand, glycogenolysis is providing us the glucose, we don't need, stop it. Lipolysis will provide the free fatty acid, we don't need, stop it. Gluconeogenesis will give us the free glucose, we don't need it, stop it. And lastly, the ketone body synthesis will also be inhibited by the action of insulin. So any enzyme which is there in these pathways will be activated by insulin. What this means, all these enzymes are active in dephosphorylated form. By insulin causes dephosphorylation, whereas all these enzymes are inactive in the dephosphorylated form. Nobody is going to tell you like this so that you can quickly remember. So insulin causes dephosphorylation. The pathways which are down-regulated by insulin, there the enzymes will be inactive. So in dephosphorylated form, those enzymes are inactive. When you do the phosphorylation, those enzymes will become active. On the other hand, in these pathways, when you do the phosphorylation, the enzymes will become inactive. So in this way, you can quickly remember which enzymes are active in the phosphorylated form, which enzymes are active in the dephosphorylated form, and the net action of the insulin. Okay, so please keep this point in mind. Very important concept. Repeatedly we'll get the question from one of these 4 points which I have mentioned. What is happening in the fed and fasting state? Which enzymes are active, which are not active? What is happening when there is increased insulin? What is happening when there is change in insulin glucagon ratio? What enzymes are active in the phosphorylated form? What enzymes are active in the dephosphorylated form? Going back to the question, see here, low insulin glucagon level. In other words, we are dealing with the low insulin level. Which enzyme activity is increased in low insulin level? In other words, which enzyme activity is decreased by insulin? So you have to identify which enzyme is inhibited by insulin. Hexokinase, glucokinase, part of glycolysis, they will be activated. Pyruvate kinase, part of glycolysis, they will be activated, whereas glucose 6-phosphatase is part of the gluconeogenesis, which is inhibited by insulin. So when the insulin level is low, this enzyme activity will increase. This is how we are going to attempt the answer. I hope I have been able to give you a very important concept. 4-in-1 questions. I have discussed 4 questions using a simple question, and one of these 4 questions will definitely be there in your exam. Without fail, one of the questions will be there in your exam. Okay. Now I've used a clinical case to again discuss a very important topic. Almost always will get the question from that topic which I am going to discuss. 4-year-old boy of first degree consanguineous couple. Why the question mentions consanguineous couple? Because here in these cases the chances of autosomal recessive disorders will increase. Was noted by the parents to have darkening of the urine to an almost black color when it was left standing. Please note, it becomes black when it is left standing. Otherwise it will be normal. It will become black when it is left standing. He has normal siblings. There are no other associated medical problems in the patient. No other signs and symptoms. Growth and development are also normal. Which of the following enzymes is likely deficient in this patient? Very good. So I'm sure all of you have been able to make the diagnosis that this is a case of L-captonuria. This is a case of L-captonuria where the deficient enzyme is the homogentisate oxidase. Now this disorder is a part of the cluster of phenylalanine tyrosine metabolism where we have a series of disorders. So let us quickly revise the disorders which occur due to the phenylalanine tyrosine metabolism defect, and very quickly we'll take a look at the enzymes which are involved there. Okay. So we start with phenylalanine, which is converted to tyrosine by the enzyme phenylalanine hydroxylase. The deficiency of the enzyme, when the enzyme is decreased, in that case we'll get the phenylketonuria. This particular reaction also requires the coenzyme. The coenzyme is the tetrahydrobiopterin, which gets converted to the dihydrobiopterin, and it is then recycled to give rise to the tetrahydrobiopterin. The tetrahydrobiopterin is synthesized within the body. So 4 different enzymes are involved in the metabolism and the recycling of the tetrahydrobiopterin. If any of them is not present, in that case the tetrahydrobiopterin level will also fall and we get again the phenylketonuria. This is described as the malignant type. Tyrosine is then converted to the hydroxyphenyl pyruvate, and the enzyme which is required here is the tyrosine aminotransferase. The tyrosine aminotransferase will convert tyrosine to hydroxyphenyl pyruvate. Deficiency causes the tyrosinemia type 2, also known as the oculocutaneous tyrosinemia. Because of the presentation, it is also known as the oculocutaneous tyrosinemia. In the past, it has also been known as the Richner-Hanhart syndrome. Some amount of mental retardation can be there, just like in PKU, we can have some mental retardation here also, but it is not profound. Unlike the phenylketonuria or the tyrosinemia type 1, mental retardation is there, but it is mild to moderate. The hydroxyphenylpyruvate is then converted into homogenetic by the enzyme hydroxyphenylpyruvate oxidase. If this enzyme is not working, we get the tyrosinemia type 3. This tyrosinemia type 3 is almost asymptomatic. Most of the patients will not have any signs and symptoms. Rarely they can have the mild mental retardation, but that will be all. However, in some patients, this enzyme may be defective. In some individuals, this enzyme may be defective. Alright, in case of the defect, the complete metabolism of the hydroxyphenylpyruvate does not occur. It gets blocked in form of an intermediate. So please note, if the enzyme is deficient, in that case we get the tyrosinemia type 3. This is different when the enzyme is defective. When the enzyme is defective, okay, so there are two different scenarios, whether the defect in the enzyme is there or the amount of enzyme is decreased. Homogeneous is then converted into the fumarate acetate state by the enzyme homogeneous oxidase, which we were just discussing. And here we get the condition known as the alkaptonuria. The classical finding, as we mentioned in the question, is that the normal urine is there, but on standing it becomes black. No other sign symptom will be there in the patient. The growth etc. is absolutely normal. The fumarate acetate state is finally and ultimately converted into the fumarate and acetate state by the action of fumarate acetate state hydrolase. Deficiency of this enzyme will cause that tyrosinemia type 1, also known as hepatorenal tyrosinemia. One of your friends has mentioned Fayaz, Mr. Fayaz has mentioned this is the hepatorenal tyrosinemia. This is also known as tyrosinosis. So if you see some tyrosinosis, don't get confused. Tyrosinosis is same as the tyrosinemia type 1. And here we will have significant mental retardation. In addition to the boiled cabbage odor in the urine, you find the boiled cabbage odor in these patients. Boiled cabbage odor in these patients will be there. Characteristic urinary odor is there in patients of PKU also. I am sure all of you already know what is the characteristic odor in the patients of PKU. In patients of PKU, we have the mousy or musty odor. Mousy or musty odor. Okay. So these are some of the very important disorders and the names which you have to keep in mind. Okay, very important cluster that we just discussed. Very good. Now we have another very important you can say question, or rather a point which is commonly asked. See, lipid metabolism is very vast, but from there we get very specific questions. Mostly they will talk about the transport of the lipids, where the lipoprotein will come into picture. In lipoprotein, the proteins which are there are known as apolipoprotein, which help us to distinguish between the different types of lipoproteins. So question which is asked is apolipoprotein integrated in HDL are all except. So you should know the B family is not there in HDL. Everything else will be there in HDL except for the B family, whether it is B48 or it is B100, it will not be there in HDL. Everything else, A1, A2, A3, and the ApoC, ApoD, ApoE1, E2, E3, E4, all of them, sorry, no ApoD, ApoC, ApoE1, E2, E3, E4 will be there in HDL, but B48 and B100 will not be there in the HDL. In this context, what you should know is the function of the apolipoproteins. This is also very frequently asked. What is the function of the apolipoproteins? So let us take a quick look. Function of apolipoproteins. Number one, they can act as enzyme cofactors. They can, ApoC2 will activate the lipoprotein lipase, the ApoE1 will activate the lecithin cholesterol acyl transferase. They can act as enzyme inhibitors. The ApoE2 and ApoC3 will inhibit the lipoprotein lipase. The ApoC1 will inhibit the cholesterol ester transfer protein. And lastly, they can act as ligand for interaction with the lipoprotein receptor in the tissue. B100 and ApoE will interact with the LDL receptor. The ApoE will interact with the LRP LDL receptor related protein, and ApoE1 will interact with the HDL receptor. So these are some of the important functions of the apolipoproteins, very commonly asked in your exam. Okay. An associated clinical case which is commonly asked here is the Zellweger syndrome. Zellweger syndrome. This is the condition where the peroxisome is either absent, or normally it has a lot of enzymes. There are no enzymes in here. So if peroxisome is either empty or it is not there, in this case we get the Zellweger syndrome. Zellweger syndrome is also known as the cerebrohepatorenal syndrome. It is known as the cerebrohepatorenal syndrome, and here there is accumulation of very long chain fatty acid. Very long chain fatty acid will accumulate, giving rise to the cerebrohepatorenal syndrome or the Zellweger syndrome. Okay. Then we have this question which talks about the breakdown of the purines where the uric acid is formed. All of the following enzyme deficiency give rise to hyperuricemia. Most of these will be the inherited disorders. So here there are some conditions where the enzyme deficiency will cause hyperuricemia, but at the same time there are certain enzyme activities which when increased will cause the hyperuricemia. So you can see glucose 6 phosphatase decreased hyperuricemia, AGPRTase decreased hyperuricemia, aldolase B decreased hyperuricemia, but xanthine oxidase when it is increased it will cause hyperuricemia. Okay. Also the PRPP synthetase when it is increased it will cause the hyperuricemia. I'm sure you know the relevant diseases. Glucose 6 phosphatase deficiency is the von Gerke's disease. The AGPRTase deficiency is the Lesch-Nyhan syndrome, whereas aldolase B deficiency is the hereditary fructose intolerance. Right? It is the hereditary fructose intolerance. So in all these conditions, decreased enzyme activity is resulting in increased formation of the uric acid, whereas in case of xanthine oxidase and in case of the PRPP synthetase, the increased activity will cause the hyperuricemia. So here your answer will be xanthine oxidase. This is a trick question, very commonly asked. You should know the two enzymes where increased activity will cause hyperuricemia, and all others the decreased activity is going to cause the hyperuricemia. All of them are inherited conditions. Okay, all of them are inherited conditions. Okay, very good. So here, very briefly, I'll talk about two things. Number one, the hyperuricemia. What are the causes for the hyperuricemia? So when you talk about the hyperuricemia can be occurring due to two different systems getting involved. One is metabolic. Metabolic means there is increased synthesis. Second is the renal system. Renal system means there is decreased excretion. So any of the two systems, when they are defective, they will cause hyperuricemia. In metabolic, some of the causes are inherited in nature, which we have already discussed. We just mentioned the 5 enzymes. Some of them cause hyperuricemia when they are increased in activity. Some of them will cause hyperuricemia when they are decreased in activity. Whereas there are other causes which are metabolic in nature but not inherited. We call them the secondary metabolic causes. Here, what all you should know? Any myeloproliferative syndrome, leukemias, where the cell turnover is increased, any cancer, psoriasis. What is happening in all these cases? The cell turnover is increased. When the cell turnover is increased, the degradation of the nucleic acid will be decreased, DNA will be increased, and more of the uric acid will be formed. So any condition which causes increase in tissue turnover. Sometimes there can be dietary causes also. For example, if there is excess fructose in the diet, if there is excess liver in the diet of non-vegetarians, or if there is excess consumption of beer. All of these are dietary causes for hyperuricemia. These are the dietary causes for hyperuricemia. Talking about the renal causes, most important and most common would be the chronic kidney disease, which can then progress to the end stage renal disease. In some individuals where there is prolonged use of the NSAIDs, the NSAIDs will cause damage to the kidney. We know that they are nephrotoxic, so there the function of secretion of the uric acid can be affected. Lactic acidosis, because of any reason, most commonly it is due to the acute alcohol intoxication. So what happens, lactic acid will compete with uric acid for the excretion. Both of them are being passed out by the same pump. So if there is more and more of the lactic acid, then the excretion of the uric acid will be decreased. Then there can be certain drugs which can block the pumps, like the thiazide diuretics. The thiazide diuretics will inhibit or affect the pumps which are causing the excretion. And lastly, some toxicity can be there with heavy metals. For example, the lead toxicity. The lead toxicity has its own name, commonly known as the Saturnine gout. So hyperuricemia due to lead toxicity is known as the Saturnine gout. So these are some of the very important causes for hyperuricemia, very frequently, very commonly coming in your exam, which you should know. You should be able to identify some of the very important causes and the relationship to the increased uric acid. Okay, in the topic of the nucleic acid, one very important point which I want to talk to you about is the polymerase chain reaction. The question can come in multiple ways. The polymerase chain reaction is very important for our exam, keeping in mind the COVID pandemic. You know that the diagnosis for the COVID, the gold standard which has been used during the pandemic is the RT-PCR, reverse transcription polymerase chain reaction. So we should know the basics, the fundamentals of the polymerase chain reaction. It is one of the most likely questions to land up in our exam. Okay, so about polymerase chain reaction, what you should know? What are the substrates? What are the substrates which are required for the polymerase chain reaction? So here, the substrates which are required are the template DNA, the primers, the polymerase. Typically the Taq polymerase is being used, and the substrate. Substrate is the deoxy form of the nucleotide triphosphates. Please note there is another molecule called DDNTP, dideoxy nucleotide triphosphate. The dideoxy nucleotide triphosphate is not used for the polymerase chain reaction, rather it is used for the Sanger technique, where we are doing the sequencing. It is used for the Sanger sequencing of the nucleic acid. In addition to this, we need the magnesium as the cofactor and buffer to maintain the pH during the reaction. Second part which you have to know is what are the steps of the polymerase chain reaction. The steps of the polymerase chain reaction. Number one is denaturation. You have to separate the two strands of the DNA. So number one is denaturation. Once the two strands are separated, the primers are allowed to join, which is known as annealing. And lastly is the elongation, sometimes also known as the extension step. These are the three steps which are required for the polymerase chain reaction. Before you carry out the polymerase chain reaction, primer synthesis has to be done. And after the elongation, we run it multiple times. 35 to 40 cycles are there. Once all the cycles are there, we also have a final elongation step, which is much longer. It is not part of the cycle, but separately we have to do the final elongation step. So these are the very basic about the polymerase chain reaction that you must know. What are the substrates for polymerase chain reaction, and what is the sequence of steps which is required to carry out the polymerase chain reaction. We start with the synthesis of primer, then denaturation, the annealing and elongation, which runs multiple times, 35 to 40 cycles, and finally the final elongation step when the PCR is completed. The PCR is done with DNA. So if you're dealing with RNA, this RNA has to be first converted into DNA. Typically it is converted into the complementary DNA. This process is known as the reverse transcription. Once the cDNA has been formed, it can be used for the polymerase chain reaction. The whole process is known as RT-PCR. So please note, the technique used for identification of the COVID virus during the pandemic was the RT-PCR. It is a two-step process. First, we carry out the reverse transcription. Using the RNA, we make it into the complementary DNA. Then we use that complementary DNA for the polymerase chain reaction. So RT-PCR means reverse transcription followed by the polymerase chain reaction. This was used as a gold standard for diagnosis of COVID infection during the pandemic. It's still being used for confirming the diagnosis of the COVID. Okay, so these were some of the important points and some of the very important questions which I think are supremely important for our exam, which is just around the corner. So I hope you are able to benefit from these points that we discussed. Please note that now don't read anything new. No new topics. You are only going to revise what you have already covered. If you start reading new topics now, then whatever you have read, that will also be a lost case because you will not be able to revise. So second part is try to optimize your revisions. More revisions you have, the easier it will be to recall the topics and the information in the exam, and higher chances of marking the correct answer. Okay, and there must be the revision of high yield topics in last 3 days all of us are continuously harping that is very important, high yield, high yield. So those are topics which should be revised again before you go for the exam. So these are three important messages for you which I want you to incorporate in your preparation in these last few days to get a good result. So I would say the very best of luck from my side. Don't lose the concentration at this point in time. Stay focused and you will come out with flying colors. Once again, best of luck and thank you very much for attending this session.